I realize as I'm sitting here listening to other people give their talks that I chose the least mathy talk I could have given. So I apologize for that. This is going to be a little more high level, more modeling concepts, things like that. But if nothing else, it'll give your brain a little bit of a break from all these equations you've been seeing, because I'm not sure I even put any in here. All right, so one of the reasons, though, that I wanted to talk about this today is I'm going to be talking about network dynamics in a potentially different way than a lot of people here have been talking about. And I'm going to be talking more about how the structural network changes. Okay, so we're not going to be thinking, I'll touch briefly on how this would impact actual brain dynamics, but we're going to talk more about dynamics as in dynamics of the network, where the actual connections in the network are changing, and what that can mean, and the implications that has when you're looking at something like traumatic brain injury. And another reason I wanted to talk about this today is this is all work done with Dr. Dave Polson, who was at UB, and he unexpectedly passed away this summer. So I'm going to show you what we've been working on, what we've been able to publish, where we wanted to go, and I honestly, I don't know at this point how much of this work is going to be able to go forward, because he was one of the few people that can actually do the experiments I'm going to be talking with you about today. So we have a grant under review right now. We're trying to see if we can find another replacement, but he was a great person, and so I want to make sure that we do talk about some of the work that we've been able to do, because we were really excited about where this was going. All right, so the first thing I need to tell you about is a little bit about traumatic brain injury. All right, this is actually a huge problem. So here's just an example from the United States. It's the leading cause is one of the leading causes of death and disability in the United States. All right, and there's a lot of people that are dealing with some sort of traumatic brain injury, and falls are actually the leading cause of injury. You can see here that's 35.2%. But there's a lot of, whoops, okay, gotta get used to this thing. All right, there's a lot of different types of injury one can have to the brain, and that's going to be important in some of the things that we're talking about, is that it's not just like traumatic brain injury is, a thing that you can define. There's many different ways to receive some sort of injury to your head. All right, and we think that that's important. So one of the reasons that we're really interested in studying this is actually about 20% of people who receive some sort of traumatic brain injury will go on to develop epilepsy. Okay, so I've been working in the field of epilepsy for a while now, and so this is really interesting, right? So there's this idea of some people who get an injury will develop epilepsy, some people aren't, and there's really no way to predict when that's going to happen. All right, you might think it's the severity of the injury or something like that, but there's no way that anyone actually knows to predict who's going to develop epilepsy right now. And sometimes this happens, relatively soon, 6 months after the injury. Sometimes it's 20 years later. Right? But the idea here is that when people start thinking about this, you have the injury, sometime later you progress to clinical manifestations of it. But the hope is that in between there's some sort of window of opportunity where we could intervene if only we knew who was going to actually develop epilepsy. And epilepsy isn't the only possible outcome, okay? One of the interesting things about traumatic brain injury is that there's a whole range of possible outcomes. There's cognitive defects, there's sleep disruptions, all kind of things can happen. And again, we don't really have any good biomarkers for determining when this might happen. All right, so we need to find these biomarkers. And so some people have looked at this. There's some electrophysiological data out there. People see maybe high-frequency oscillations that start happening in people, and that makes them more likely to develop epilepsy or to have some of these more severe outcomes. People can look at sleep spindles. But every time someone finds one of these, another study says it's not true, they can't find it, and they find something else. All right, so this is a huge problem. And there's also conflicting data if you look at animal models versus human studies, okay? And one of the reasons that this is so difficult to study is, like I said, it's not that every injury is the same, right? So if you are looking at human data and actual clinical data, you have this problem of, okay, you have this database of all these people that have been classified to have received some sort of TBI. Well, how do you figure out where they actually got hit? What was involved? You know, like there's so many questions, and there's so much heterogeneity in the type of injury that you think, okay, well maybe that's what it is, right? That's what's leading to this. It's just that it has to be a very specific type of injury, and there's all these differences, and we just haven't figured out what that is yet. But that doesn't seem to be the case. So what a lot of people have done looking at this is, the other problem is if you're looking at human data, right? You can't just like go open up someone's brain and make little slices of it and do the histology and figure out exactly what happened, right? So you have to use these non-invasive imaging techniques. And so a lot of people have looked at fMRI, all right? So they take someone, they put them in a scanner, and they look at this functional connectivity, where you're looking at brain regions, and you're basically asking, you know, what's the pattern of synchronization in the brain, and trying to understand differences in this fMRI data. So what I'm going to talk about, though, is a little bit different, because we know that the dynamics of the brain are impaired after some of these injuries, and you'll see that in the functional connectivity. But the dynamics of the brain happen because of this underlying structural network, and there's actually a lot of structural connections in the brain that are changed as a result of injury. All right, and this happens across all types of different scales, down, you know, from the little neuronal level and things like that. What I'm going to talk about is more of these large-scale brain networks, where we're actually interested in white matter tracts. So these are connections between brain regions. All right, and even at that level, we see a lot of disruption in the network. The way people normally study this, though, is to look at this MRI imaging, and they look at these measures like fractional anisotropy, mean diffusivity, radial diffusivity, and those are just kind of measures of how things are happening. All right, you can think about it as like a map, and you'll just have, for each pixel volume, you'll have a measure of the fractional anisotropy and things like that. So these are properties of how water molecules are diffusing in your brain, basically, that try to tell you something about the structure of these tracts at that point. And so I'm not going to get into what these measures mean, but they're not really thinking about the brain as a connected network, okay? But what people do know is that we have specific changes that are heterogeneous, all right? And that even if you have some sort of local injury, the changes aren't happening only at the local level, right? They're happening at the more global level across the brain. And so what we're interested in our work is trying to think about this as more of a network problem, okay? So some people have looked at the structural network changes in TBI, but again, like, these are all kind of conflicting. They're mainly done in humans. So there's this whole problem of, you know, the heterogeneity of the injury and how that might affect these changes that you see. But in general, people see changes in the hyperconnectivity. If you look at the betweenness of the network or the eigenvector centrality, you'll see differences after injury. There's increased diffusion times. But it's just so difficult to figure out what's going on, because all of these studies are done in these human populations, and you have all the factors of age, family history, depression, alcohol use, the injury severity, the location, the type of injury. Because remember, TBI, we're not even talking about the same type of injury to the brain. Forget just the location. There's a lot of factors that go involved. So what we're interested in doing is trying to remove a little bit of this, and instead using an animal model of traumatic brain injury. And so this is the model that my collaborator Dave worked on and was very good at. And so I was telling some people about this at lunch the other day. Like, it's kind of horrible. What they do, this is a lateral fluid percussion model. So you can see you have your rat there, it's head fixed, and you pull up this little lever to a certain angle, you swing it, and that induces a pressure wave, which travels through this tube and induces an injury, okay? So the good thing about this model is that you have a very controlled model of injury now, all right? All of these rats are receiving the same injury. You're using the same pressure, just the angle that you pull this up to controls the pressure, and you can measure the pressure that's being put through there. And as you might imagine, how high you pull up that lever changes the severity of the injury. Okay, so you can vary that. And because you're head fixing these animals, you are, in theory, hitting the same place in each of them. Okay, there's very precise ways for measuring this on a rat's head so that you know where you're going. So the hope is that if we look at this model, we're eliminating some of these extra effects that are out there in terms of the location and type of injury, and we can really just then talk about, okay, what are, from the same injury over and over, what are the types of network changes that we see? What's really interesting about this model is that there's still a huge range of different outcomes that happen from the result of the same type of injury, okay? So what my collaborator would do in his lab is he would actually assess these using a neurological severity score, and it's based on a whole range of motor tests and sensory tests, if they have seizures at the time of the injury, things like that, all right? And so if you look at, this is the score here, and this is the pressure that the animal's hit with, okay? So in general, things down here are going to be, in the traditional words of this model, called mild injuries. All right, but these things up here beyond 3, those are severe injuries, okay? So what most people look at are these severe injuries in these models, well, not most. One of the things people look at, and what we're looking at here is a severe injury model where they're receiving these higher pressure injuries. But if you look at this neurological severity score, you still see there's a huge range of outcomes here. All right, some of these, and remember, these are motor tests and sensory tests. They're doing water mazes and, you know, all these kind of weird things they have animals do. And so when you score them on that, they're all performing very differently, despite the fact that in theory they've all gotten the exact same injury, okay? And this is really interesting to us. All right, one of the things that people complain about this model of epilepsy is that so few of them go on to develop epilepsy. Like there's about like 30% of these go on to develop epilepsy, which is interesting in and of itself, right? But what my colleague actually found was that if you classify this and you do this based on their neurological severity score, and you only take the ones that have what he classified as a severe injury, then about 60% of them will go on to develop epilepsy. So there is some sort of correlation here between how they're performing, and this is, I should say, this is all done, like, I think 24 hours after the injury, okay? So there's some sort of correlation with how they're performing immediately after the injury and what will go on. But it still isn't exact. We're still talking 60% of them. Not all of them get epilepsy, and no one understands why. All right, so we set out to say, well, can we try to figure out why this is happening if we, instead of thinking of this as just, you know, like, oh, there's a local injury, it's happening here, try to think of this as a network, okay? So our idea was, based on this structural brain network, can we predict who's going to develop PT? This is post-traumatic epilepsy. So this is a rather small data set. You know, this is a hard model to do, and there's actually a fairly high non-survival rate. But what we did is we performed what's called DTI. This is a diffusion tensor imaging, 5 weeks after the injury. And what we did is we extracted the tractography to get these structural brain networks. So just a very brief thing into this in case you've never heard of this. This is a non-invasive form of imaging. And what it does is it's an MRI technique, and you have a magnet, and you use different angles and different strengths of the magnet to take these images. And then people, not me, have developed these wonderful algorithms that track the diffusion of water molecules and how these strengths and angles change all of that. And what happens is when you have these white matter tracts, water is going to diffuse along them. Okay, so by looking at how the water is diffusing through all of these things, there are these very fancy algorithms that we hit the button and use that tell us where these tracts are in the brain. Okay? And so what you get is something that looks like here. This is 2 different views of these white matter tracts. So these are these large axon bundles that connect brain regions. All right, so we're not talking about, you know, damage at the level of neurons or things like that. We're talking about these large, long-range connections throughout the brain that are getting damaged. All right, and that's kind of important when you think about everything I'm going to talk about, too, is the scale that I'm talking about. Okay, we're really talking about these larger scale effects that a lot of people just assumed wouldn't happen if you have some sort of local insult. All right, kind of the traditional way of thinking about this is you have a very local injury, you're only affecting, you know, maybe the immediate tissue right there, and not thinking that this might have some sort of global effect. And I'll show you that's not actually what we see. So to build these networks, we take these little tracts, And we superimpose an atlas on the brain. So this is a brain for our rat atlas that someone came up with. These are basically anatomically defined regions. We have 150 of them, 75 on each side. And then you count the number of tracts connecting each region. Okay? We actually normalize it by the sum of the volume of the regions to try and turn it into some sort of density to correct for the difference in size between them. And so what we end up with then is we have our tracts, we put our atlas on, and we get our network out. So our network is, the nodes are these brain regions, and the edges are the density of fibers connecting them. Okay? So it's kind of the strength of these long-range connections between brain regions. So then it's great. Once we have a network, we can just throw a whole bunch of network statistics at it, right? And we can try and analyze it. And so the first thing you might think is like, okay, well, we have some rats that we didn't injure. We have some rats that we did injure. Let's figure out where things differ, right? Presumably we'll see some sort of difference that the injury has caused in the networks. If you just look at the adjacency matrix that you get out of this, and you look at the similarity, here this is dissimilarity, you don't actually see any significant differences between networks in the control group and in the injured group. Okay, this isn't a metric that actually allows us to differentiate which animals had an injury. Forget this whole thing of the spectrum of injuries, right? And we looked at the eigenvalue spectrum too. Again, we could not see any differences between control and injured. So that was not good, right? Because if we want to study the variability within the injured group, we better be able to see a difference between the injured and the control, right? So we looked at some other things, and we said, okay, well, looking at the full network is not giving us anything. Let's look at some subnetworks. And so what we looked at is this network-based statistic, and this identifies connected components in which the edges increase or decrease in strength. Okay? One of the problems in doing this type of work is multiple comparisons. So this is a technique that people developed to try to get over that problem of multiple comparisons in these networks with tons of pairwise connections. And so what we found is that if we looked at this, we said, okay, well, what we're seeing is that there are some of these connections that are getting strengthened, and there's also this other smaller network where the connections are getting weakened. Okay, so we have this shift towards stronger connections, and in a few places we're seeing a shift towards smaller, or towards weaker connections. And then we also looked at how that varied with distance. And you can see that we're seeing an increase in these shorter range distances and a decrease in the longer range ones. So we said, okay, well, we don't really know how to interpret that. So let's look at this minimum spanning tree. So we said, okay, if you look at these, we see differences in basically, this backbone, the core, the minimum spanning tree of these networks. And we also then looked at this trying to apply a cost function where we took this distance into account. And what we do is we see this shift towards weaker and lower cost connections after TBI. So at least we know something is happening in these networks, okay? There are these changes, we can view them. Looking at just like the network as a whole, we don't really see them, but we can start coming up with different measures to try to tease out, how we can measure these differences. And so if you look at this, then we also started looking at, just like some standard network things that you might think of looking at. We looked at the degree, we looked at the betweenness centrality, we looked at the local efficiency of these brain regions. And they show certain brain regions that do have differences between the control and the injured groups. But if you see here, the red increase, the blue decrease, and these are just the location of the brain, the regions that this happened to on the brain. But we don't really see a pattern here in what's going on. One thing I should point out, the injury is like right about here. Okay, so over here, it seems like they may be in the efficiency of the network. There's something maybe in the degree. We see a decrease here at the point of injury, but we also see these increases. But this is interesting because what this tells us is that there's something way more complicated going on than what most people thought. This isn't really a local injury, all right? And we're looking 5 weeks after the injury. So this is also surprising to people that you have these large-scale changes in brain structure in a relatively short amount of time after the injury, okay? All right. So we've realized that these local measures were telling us a little more. So we looked at motifs. Or I should say my grad student decided to look at motifs. Anyone who knows me knows I hate working with motifs, mainly because I feel like they're so subject to noise, right? You have something wrong in your network, you have a little extra edge, and that changes the motif structure. However, it turns out I'm going to be talking a lot about motifs today because this was really important for what we looked at. So we looked at some standard three node and 4 node motifs. And I should mention these are undirected networks. When you do the tractography, you have no idea how to tell which way signals are flowing on these tracts. You just know a tract exists between 2 brain regions. Okay, so we looked at these and we looked at their motif coherence. So this is just looking at how much a region participates in a given motif, all right? And these are weighted networks, so this is a weighted measure. So if you look at this, here's an example of just a 3-node motif and a 4-node motif. So these are the different brain regions down here, just looking at its motif coherence, and they're sorted based on the control group. Okay, so you can see the line, here's the control going down here. And then in the TBI group, you see this, they're kind of all over the place. Some go up, some go down, okay? And it's different for each motif, all right? But what we're seeing is that there's these large changes happening at this local level in terms of the motifs between the TBI and the control. And when you think about this in terms of noise, like when we're looking at the control group, we have all the same problems of noise kind of in both of these situations, right? We're doing the same imaging technique on both of them, the same tractography. So it's unlikely that this is really just, imaging effects that we're seeing. And if you map these onto the brain, you can see that there's kind of this weird spatial patterning. And over here, this is the point of injury. You can see we have, I should say, this is a very confusing figure. This is looking across all motifs. We tried to sum up a whole lot of things in one figure, which might have been a mistake. So let's see, the color is the z-score. And the size of the circle is the percentage of motifs that had a statistical change. Okay, so in some of these that are smaller circles, maybe only one motif went up, all right? But these big red circles means a whole lot of the motifs went up in their motif coherence, if that makes sense. And so you can see that maybe there's something associated to the site of injury, and then there's other places where it's going down. So what this tells you is that there's all of these little local changes that are affecting how often these brain regions are participating in these motifs. And it's not limited to the point of injury. It's happening across the brain, and we don't really understand what's going on in terms of this. Okay, so I told you that we're not really just interested in trying to detect which one of these animals had an injury, right? That's not meaningful to us. We want to know who's going to develop epilepsy. So we're really interested in looking at the injured group and trying to understand what the differences are in the injured group, okay? So now that we have some things that are telling us, we have some idea of what these measures are telling us, we wanted to look just within the TBI animals and try to figure out if we can identify meaningful changes that might differentiate between the severity of the injury and the clinical outcomes, okay? So this is, again, another rather confusing plot. So if you look at this, these columns here, this is a single network measure. So this WEI, that's the weighted adjacency matrix. So this is if you take within the TBI group and you just do k-means clustering on it. Okay, we varied the level of k and used the silhouette method to select the most significant one. What this tells you is that if you use this to cluster this just based on the adjacency matrix, you're going to get 2 groups. Almost all of them are going to be in one group. You have one that's tossed out into another group. Okay? If we use the eigenvalues, we actually saw the same thing. Okay, and then, so then we look over here, you see the NBS. All right, so this is that network-based statistic. These are these subnetworks of increased or decreased connections. we see a different clustering. So now there's still 2 groups, but we have animal 4, 7, and 8 in one, and the other split into another. Okay? So basically what we were trying to do is say, like, when we measure something, can we break this into like little groups, okay, for each of these measures so that we could somehow define differences between them. So if you look at this, what you notice is that for a lot of these, we don't really see any sort of similar group structure, but every time we look at these motifs and the changes in motifs, we see it's great. Well, this one, there's 3 groups. This yellow is a third cluster, but it's breaking into 2 groups, and it almost always has the same group assignment when we're looking at the motifs. So what we did then is, this is just kind of this initial analysis of saying, are these measures separating out different types of things? But then what we can do is we can actually build what's called an association matrix, and we say what fraction of times are the animals in the same group across all of those different measures that we looked at. Okay, and so then you can cluster this association matrix to say basically which animals consistently had the same types of changes, okay, when we're looking for things. And then we clustered that just using standard modularity maximization. And what happens is you get out that there are 2 communities in this network, okay? They are driven by this change in motif. Like, I mean, just looking at this, you can say, okay, well, these are always going to be, in the same group. But it's really these motifs that seem to be differentiating. And there's potentially some other structure going on here. We have animals, what is this? 4, 5, 6, and 7. No, 1, 2, 3, 4, 5, 6, 7, 8. Maybe you can see like some sort of hierarchical clustering going on there, but it's a small data set, right? We're working with what we can. But we really see that it splits really well into this T1 group. I don't really know why we chose T1 and T2. So we call them T1 and T2. And it's really driven by how their motif structure is changing after the injury, okay? And I just want to point out, if you do the same type of analysis to the control group, you do not see this, okay? This is the exact same plots. Notice that there's basically always, it splits into 2 groups. That's just the nature of these algorithms, okay? But we actually see no groups identified if we just apply modularity maximization here. They're all lumped into the same group. So we don't see this splitting in the control data. We only see this in these injured animals. All right, so we said, okay, well, let's go back to the motifs. It's clearly the motifs that are driving this grouping. And so what is the difference between how these motifs are changing in these 2 subgroups that we identified? And so if we look at the changes, these relative control, and we look at this T1 subgroup and ask how, if we look at animals only in that compared to the controls, what happens? And we take the T2 subgroup and we say, well, let's look at only animals in that group and compare that to the control. So when we split it like that, you see that now we see a very different picture than what we saw before, okay? So before we were basically taking all of these animals together and comparing them to control. So we're averaging across all the animals within that group. If we separate it out into these 2 subgroups, now what we see is that, okay, actually in this subgroup, we see a whole bunch of regions that are increasing their motif coherence. Okay, so they are participating in many more of these little local patterns, these 3 and 4 node motifs. Whereas in the T2 group, we don't see that at all. We actually see decreases across the brain. There's a few that increase, and this is close to the spot of injury. That may or may not be meaningful. Okay? But we see a very drastically different pattern in what's going on, right? And that's why we're seeing this separation into these 2 groups, is that however these networks are changing, they're doing it in very different ways, okay? So this is what we had before when we lumped everything together. And because you're averaging across these, you can't see the effects, okay? And this is a huge take-home message here, is that what most people do when they analyze this data and they look for changes in network structure, this is true across all forms of medicine, is you take your injured group, you take your control, and you compare them, all right? But you can see here that does not make any sense in this setting because we're completely washing out the effect that there are actually subgroups within that injured group. All right, and I think that this message here really could explain a lot about, I don't know, the whole world of medicine and how we think about looking at things like injury and disease and things like that. Right? You can think right now, we're living through a pandemic. There's all of these studies going on trying to determine what drugs work to treat COVID, right? And trying to determine whether or not they reach, statistical significance in terms of having an effect. But you're assuming there that every single person who gets COVID has, like the same mechanisms involved or the same reasons that they're having these symptoms or things like that. And by assuming that your diseased group or your injured group is homogeneous, you're not going to see these effects, right? So maybe it's highly effective in 10% of your population and not effective in 90%. The way that we analyze our data is not going to see that. And that's what we're seeing here, is that the way that people are traditionally analyzing this data is to assume that you have, something that's, basically Gaussian, and you have a bimodal distribution. That you can't make that assumption that the mechanism is the same for everyone in your injured group. And I think that's a really important thing that we're seeing here, is that we have two very distinct paths that these animals seem to be going down here in terms of how their brains are reorganizing, okay? And I say two, this is a very small study, right? We only had 8 injured animals. I should mention these are very expensive studies to do, to stick these animals in the MRI, so it's hard to get data. But you can imagine that if you had a larger cohort, it's not maybe just 2, right? You can have kind of like all of these different paths that these animals are going down that lead to different behavioral outcomes. And if you don't have a way of separating out which path they're on, it's going to be very difficult to make any predictions because you're averaging and you're not going to see that they're actually, I like to refer to these as network trajectories. You're essentially, going down some sort of trajectory, and how do you tease out which trajectory you're on, all right? Are you going to be someone whose brain is reorganizing like this or someone whose brain is reorganizing like this? How do you determine that? So we're thinking about this in terms of epilepsy, so we wanted some way of relating this to, would these rats develop epilepsy? Unfortunately, like I said, these are expensive studies. My collaborator had all kinds of data, but did not have a single data set where he had all of the things together. So in other data sets, we know which animals had epilepsy because they were recorded with EEG. In this data set, though, and in those we don't have the MRIs, all right? So in this data set where we have the MRIs, we did not have the EEG data. So we can't be sure about which ones would develop epilepsy, okay? But what we can do instead is we can use this network structure to predict the functional connectivity. Okay, epilepsy is defined by the spontaneous occurrence of seizures, more than one seizure. Okay, we have to have at least 2. So there's this idea that synchronization is involved, and you have this excessive amount of synchronization in the brain that allows for this spread of activity and goes to seizures. So what we did is we said, okay, well, let's estimate the functional connectivity. So people have looked at this, and there's multiple ways you can go about trying to estimate functional connectivity in these networks. We just took a very simple method looking at the path transitivity. Okay, so what this does is you're estimating basically the synchronization between brain regions based on these shortest paths between them and the number of reentrant connections. All right, so you can see here, this is kind of the example. You have these little kind of loops that you can go to. How many of these little loops put you back on path to being between them, okay? And so what happens when we predict this is if we look at, here we have the controls, okay? This is their simulated based on the average connectivity of the controls. Here's the simulated functional connectivity for T1, and here it is for the T2 group, okay? It is remarkably hard to see this, and I should have drawn the little line here. I always forget to add it until I'm talking. But what happens is you can see that the path transitivity, so what this is measuring, and this is relative to the controls, okay? So anything above 0 means it's greater than in the controls. And what we're seeing here is that we actually see a significant increase in the T1 group relative to the controls. Okay, so if we average them out, we don't really see anything. This is not significant. Here, however, we see this significant increase in the T1 group, and we don't see that in the T2 group. So what this is telling us is that on average, and this is a very gross measure and not the best way of doing this, but we think that what this means is that this T1 subgroup is more likely to synchronize, all right? The brain is more likely to have higher synchronization levels throughout the network. So our prediction then, oh, and I can show you where these changes are occurring too. So this is interesting. So if you look at this, that I showed it in matrix form before. This is just taking that matrix and projecting it onto the space of the brain. Okay, so now you're actually getting to see where those connections are in the brain that are increasing in their functional connectivity, in their path transitivity. So this is if we just average over them. Okay, you don't really see patterns. If we split this now into this T1 group and we use their structure to determine this, what you can see here is that there's this region over here where we see a decrease in the propensity to synchronize, but everywhere else it's increasing. All right, and these are just right, top, bottom, left. This is just flipping the brain around and looking at it in different ways. But the important thing to notice is that the injury is right about here. So what we're seeing is right around that spot of the injury, you seem to see desynchronization between these brain regions, but the rest of the brain is synchronizing. Okay? And in the other group, you see a completely different pattern, right? Here you see a lot of decrease over here, but also over here. And there's some core of connections that are strengthening, but not in the same way, not in the same spatial patterning. Okay? So we think that this is really important, but we don't entirely understand what's going on. So what we think, again, we have no idea to actually link this, but our prediction would be that this T1 group is the group of animals that would go on to develop epilepsy, okay, based on what we're seeing here. And that's about 38% of the animals, which is in line with what you might expect. Again, this is a very, very small data set, so it's really hard to say much about it. But that's kind of what we're thinking with this. But I think the take-home message here really is that we need more data on this, right? We need the full data set. And we really want to get over this idea that when we're analyzing our data, that we assume we're looking at a single population, okay? I think that one of the things that's really important when you're looking at health data and injury and disease and all of these things is not to assume that you have the same mechanism in each person, okay? All right, so this is just kind of summarizing this part of the talk. But we saw this was driven by motif participation. And so what we think is going on is that in these tractography networks, part of the problem is we have one time point that we looked at, right? So really what we need is a longitudinal data set to study how this changes over time. But what we think is happening is you're getting a lot of kind of small disruptions to the network, and you're getting a lot of local changes, right? So you have these longer tracks, and they're getting these smaller changes that are going on. And maybe this is due to the fact that we only looked at 5 weeks. We really don't know. But the idea is that's going to change a lot of these local network properties. But as that's happening at the local level, you have this insult that's changing things. The brain is trying to compensate. Okay, so you get these other changes going on in the rest of the brain too. So what we've seen here is that, when we looked at those predictions about the functional connectivity, you see a loss of synchronization near the site of injury, but the rest of the brain compensates by increasing its propensity to synchronize. And so maybe that's what's throwing off then this balance of dynamics in the brain and can lead to epilepsy, is that as the brain tries to compensate for that injury, the global effects have this larger network effect. All right, this is all pure speculation, right? The downside of this type of analysis is it doesn't tell you mechanisms, but it does give, neuroscientists and neurologists idea of actual experiments that they can go through and then try to test them. And so I will say, I told you I have a grant under review right now where we're trying to see if someone can do these experiments, but the proposal is to take a longitudinal study of this and do imaging a few animals at least to get a baseline. It's remarkably hard to convince experimentalists to do a baseline scan of the brain in this case because there is such a high death rate and it's so expensive. So you're going to do a scan and then 30% of those animals are going to die, and the experimentalist says, absolutely not, why would I scan them, and then throw them out. Personally, I study individual variation, across all different kinds of systems. So I think that perhaps one of the reasons 30% of them die is because their structure of their brain is different, and that these small structural changes that we see in people, when they get the injury, their brain reacts differently. So I'm personally really interested in doing a huge study and getting all these baseline scans. This is something you really can't do in humans, right? Because who goes to get their brain scan right before they have an injury? You don't predict that and think, I think I'll just go get my brain scan today in case someone hits me on the head tomorrow. So the animal studies are the way to do this, but again, the costs make it really hard. But hopefully someday, and there is some work going on, kind of doing some sort of similar longitudinal stuff here at UCLA and in New York, and they have this big EpiBios project. So we're hoping that maybe what we can do is talk to those people and see if they'll jump on this idea and do some experiments for us or share their data or something and see what's going on. But I think that's the real step forward, is trying to understand, how this individual variability, that's actually very hard to measure. Remember when I showed you this, like just looking at these adjacency matrices, I couldn't show you differences between injured and control. So you have to be kind of creative about how you're even detecting these differences. But the idea is that if we can do this longitudinally, we can make predictions then kind of about which path you're on. And maybe even just from a starting baseline scan, this is my dream, I'm sure it will never work this way. You go in and you say, okay, here's the type of brain structure this person has. They're likely to be susceptible to TBI, okay? They're likely to, be at a higher risk for epilepsy. You can kind of differentiate then risk levels just based on kind of your baseline structure, which is not something a lot of people like to hear. You don't want to think about this idea that, like your brain, the small changes in structure determine so much. But I think the other thing to remember is that all of this is plastic too. Your brain is changing all the time. So it's not so simple as from birth, you were destined to, have this. It's a much more complicated system than that. And these connections change based on environmental factors and all kinds of things. Okay, so that said, I think I have some more time, hopefully. Because now I want to tell you, I gave you this great story, right, about how we were thinking about this and the things involved. But there's a lot of problems with what I told you, okay? And so as we started to look at this more, we started looking at these tractographies. So when we are building these networks, okay, and hopefully you found the other part of my talk interesting. Hopefully this is going to be your real take-home message, though. All right, we are actually looking at basically strings of spaghetti in space is a way to think about them, right? Okay, you have this wad of spaghetti. It's in your brain. You can follow these fibers from point to point. All right, so that's what these pictures tell you, right? And I'm going to be showing you some more pictures of this. When we build our networks, we have our rat brain atlas. We throw this on top of there, and we count the number of connections between two regions, which seems completely reasonable, right? But that is a huge choice that we are making in how we build these networks. Okay, you can talk about the specific atlas we use, but I don't even want to talk at that level. The idea here is that actually built into the software that does this for us, people do this enough that they are just software packages you can use. When you build your networks, you can choose to build an end-to-end network or a pass-through network. All right, and what do I mean by that? Almost basically, everyone chooses end-to-end, by the way, or so it seems. A lot of people don't even put this in their methods, what they're doing. But the standard thing is to do this end-to-end. You have a fiber or streamline that goes from, point A to point B. You connect them, okay? But you can think about this. Let's say I have this white matter tract, all right? And these are my 4 brain regions. If I build an end-to-end network, region 1 is connected to region 4, okay? If I build a pass-through network, region 1 is connected to region 2. and it's connected to region 3, and it's connected to region 4. Right, these are very different networks that I'm building based on this choice of how I'm going to define connections in the network. And one of the problems of doing this is that we don't really know what happens when the fiber goes through there. You know, we don't know if it has connections to those brain regions. Like, that's not the scale that we're looking at. No one really understands this. And so we started realizing that actually this choice of how we've built our networks probably matters a lot. All right, and so before, when we were looking at biomarkers, okay, we made one choice and we see these changes, that's good. But we think that we're really missing a lot of what's fundamentally happening by making that choice of only looking at these end-to-end networks. And so what we started realizing was that if you build these pass-through networks, remember I said this injury is a local injury. You have some sort of local lesion. We know that that happens, right? You can see this on brain scans. There's a local lesion that happens. And when you have a local lesion, if that happens within one of your brain regions in your atlas, that's within the node. You're definitely not seeing that. All right, and you're really not seeing a lot of these other local changes that might be happening at that very local level. So one of the things that my colleagues found, and I don't have very many slides up here because this is a weird situation we're in now, where he was funded by a company. This data isn't mine. No one knows who controls the data. I need all kinds of permission from people I don't know to show you things. So I'm not going to show you very much, but let me tell you about some of what we were seeing. Okay, so what my collaborator found, and hopefully we will manage to get this published, is that if you look through here, this is one of these injured rats. And so this is just an example of, this is a region of the brain called the diencephalon, and it contains the thalamus. Okay, and my colleague was really interested in studying the thalamus because what he found was that they had these calcifications in the thalamus. The injury was like near the auditory cortex. Okay, and this is a deep brain structure that they were seeing these calcifications and like different sizes and things like that. They really didn't understand, and they were far away from the point of injury, so no one had really ever like thought to look there before, but they found them. And so the thalamus is just inside of this diencephalon. Think of them as the same thing. But we found that if we take this software, you can do all kinds of things to visualize it. And so we were kind of playing with trying to look at these tracts. And because he was interested in this, we said, okay, well, let's look at the fibers. What we did first is that start in one and end in the other, just between the hemispheres. So I guess I should have switched this around. So this is like the standard end-to-end. And if you say, well, show me those fibers that connect these two regions, what do I see? You see something that looks like this. That is uninteresting to me, okay? If I say instead, show me the fibers that pass through each of these regions on the right side and left side, so they don't have to start and end there, they just have to go through them, so they're connecting them in some way. This is what I see, and this is interesting to me because this is not symmetric. Okay? Remember, they get an injury on one side of the brain. And what you see here is there's essentially this hole. And it turns out we haven't been able to actually like overlay these yet. I'm trying to get this calcification data from a student now. But we think that this corresponds to where these calcifications are occurring. Okay? And so what's happening here is that when we're looking at a lot of these methods with these end-to-end, we don't take space into account. And in no way when we build our network are we taking space into account, really, right? But these are spatially embedded networks. And it seems that these fibers are being disrupted here. And remember, this isn't the site of injury. This is like a deep brain structure. But there's this hole here, and the fibers are diverted and moving and doing weird things because there's this calcification here. And that's something that we're completely missing when we look at this. And maybe that's one of these explanations for why we're seeing all this compensating in the brain, is that there are actual now physical blockades in the brain that these fibers can't go through anymore, and they're moving around. Okay? But again, if we just look at the end-to-end, I mean, like, they're not perfectly symmetric, but I never would have been like super concerned looking at this. As soon as we did this, we said, oh my goodness, what is happening? But that's really hard to find, because if I show you the picture, like, this is what you normally see when you visualize them, right? It's just like I said, it's like a blob of spaghetti. Like, it's impossible to like sift through this and try and figure out what's going on. So we're lucky that they had seen these calcifications and we had happened to go look at the thalamus to see what was going on there. And so what we think is really important is that we have to find better ways to think about space in these networks. When we have networks that are these spatially embedded networks and we're defining connections, we need to really figure out, like this is kind of a different concept than we have in a lot of networks that we build, or when we think about neurons or things like that and how they interact. But we have these fibers, we have these strings, and they're traveling through space. And the regions that they pass through are important, right? So we probably need to be thinking more about these pass-through networks if we're interested in maybe like these deep brain structures. But we also even have to think about, kind of like the spatial path of this, like how does that change? And this is a different question entirely than the type of network analysis we're thinking at. So at the network level, we're going to have to start thinking more about these pass-through networks. And then at another level, we just have to start thinking about like how do we measure properties of these fibers in space? And that's something we're not really sure how to do now, but we think that this is going to be really important as people start studying these tractography networks more and more, especially in the context of injury and disease and things like that. Is, one level of abstraction is this network level, and we can measure things in that way. But another level is going to actually be going through and figuring out kind of like the spatial trajectories of these and how that varies. And how disease maybe, change, maybe if we can measure changes in those properties, we actually might see more in these diseases because we're just not measuring our data in an appropriate way. And I think that is one of the big messages, is that for a lot of us who are not the neuroscientist or the biologist looking at these systems, we're not really prepared to think about like the actual, physical implications. But if I go talk to my colleague and I tell him something, he's uninterested in these network measures and stuff, and he says, "What's happening in the thalamus? Like, I care about the thalamus. Go look in the thalamus." And it's really important that we have that type of feedback, and that is what drives the modeling, because the step of network creation is something, again, no one talks about. Some people may even leave it out of their methods in a paper, but that's going to determine so much. And I don't think I didn't put a slide in here. If you try to look at, if you build a pass-through network or an end-to-end network and you look at how things are changed between these injured and control animals, you will be able to identify differences. So you'll say, if you just look at like something simple like the degree, right? So like how connected is each one. If you build an end-to-end, you don't see differences. If you build a pass-through, you start to see regions that show statistically significant differences. Like, we really have to start thinking about how all of these assumptions matter when we're interested, especially in these spatially embedded networks where you have these deep brain structures that a lot of things go through, and passing through there might actually be important. So that's kind of the take-home message for today. So with that, I just want to thank the people involved. Basically, everything I showed you was actually done by my grad student, Johan, not me. And then again, this was all work done with Dave Poulsen. The calcium stuff was his student, Matt McGuire. Ferdinand is our imaging person who does the MRI, and hopefully we'll be able to carry on this work and get some more of this published. So.